All right, let's get right into it. Think about this for a second. Inside pretty much every single cell in your body, and in every living thing for that matter, there's an instruction manual, a master blueprint really. It has all the information, everything needed to build and run the entire organism. And that incredible blueprint? That's DNA.
So, the big question is, what is this secret code? How does this one molecule manage to hold so much information? And maybe the coolest part, how have we figured out how to read it? Well, that's what we're going to break down.
And just to give you an idea of how much information we're talking about, get this. If you took the DNA from just one of your cells and stretched it all the way out, it would be over 2 meters long. That's probably taller than you are. And all of that is crammed into a nucleus so tiny you can't even see it. It's just mind-boggling.
Okay, so to really get how this blueprint works, we've got to start with its design. Because it turns out, the physical shape of DNA, that's the secret to everything it can do.
So the full name is a bit of a mouthful, deoxyribonucleic acid. Yeah, that's why we all just call it DNA. The best way to picture it is like a super long chain, a polymer technically, made up of these little repeating units. And this one molecule, this single type of chain, holds the genetic code for almost every living thing on our planet. Wild, right?
Now, figuring all this out wasn't a one-and-done deal. It was a long journey. It actually started way back in 1869. But the real game changer happened in the early 1950s. A brilliant scientist named Rosalind Franklin, along with Morris Wilkins, captured this fuzzy X-ray image of DNA. And that photo, that was the key. It was the crucial piece of the puzzle that allowed James Watson and Francis Crick to finally figure out its iconic shape, the double helix.
And here's where the magic really happens. It all comes down to a super simple, super elegant rule. Think of DNA as a twisted ladder. The rungs of that ladder are made of 4 chemical letters, A, T, C, and G. And here's the golden rule, the thing you absolutely have to remember. A always pairs up with T, and C always pairs up with G. That's it. That simple pairing is the secret sauce behind, well, everything.
And Watson and Crick knew right away how huge this was. This quote from their famous paper basically says it all. They realized that because A only ever pairs with T and C with G, the 2 strands are perfect complements of each other. That means if you split the ladder in half, you can perfectly rebuild the missing side on each strand. It's a built-in copying machine, a biological photocopier. Absolutely genius.
Okay, so we've got a stable blueprint, and it can copy itself perfectly. Great. But how does the cell actually use it? I mean, how do you get from a string of letters to you? That's where this core concept in biology comes in, something called the central dogma. So here's the problem the cell has to solve. The master blueprint, the DNA, is incredibly valuable. It has to be protected, so it's locked away safely inside the cell's main office, the nucleus. But the factories that actually build everything, the protein-making machinery, are out on the main floor of the cell. So how do you get the instructions from the secure office to the busy factory floor without risking the original blueprint?
Well, the cell has a brilliant 2-step system. First, in a process called transcription, it makes a temporary, disposable copy of a specific instruction from the DNA. You can think of it like making a photocopy or sending a quick working memo. This memo is called messenger RNA, or mRNA. Then, step 2, translation. That mRNA memo leaves the nucleus, goes out to the factory floor, and these little machines called ribosomes read the memo's instructions to build the proteins that do all the work.
But wait, how do the ribosomes actually read that memo? Well, they use a language, a specific set of rules called the genetic code. And it's actually pretty straightforward. The machinery reads the letters on the mRNA not one by one, but in groups of 3. Each 3-letter word, which is called a codon, is the code for one specific amino acid. And amino acids, of course, are the building blocks of proteins.
And scientists figured this out with some pretty elegant logic. Think about it. You've only got 4 chemical letters to work with in the RNA alphabet, but you need to write the instructions for 20 different kinds of amino acids. So a 2-letter code wouldn't work, right? That only gives you 16 possible combinations. Not enough. But what about a 3-letter code? Well, that gives you 64 possible combinations, more than enough to get the job done. It's a simple but powerful system.
So we've got the blueprint and we cracked the code for how to read it. That brings us to the modern era and to a project of absolutely epic proportions. What happened when humanity decided to read its own instruction manual from page 1 all the way to the end?
The sheer scale of this task was just monumental.
I'm talking about the Human Genome Project. This was a massive, worldwide effort that finished up in 2003, and its goal? To read every single letter in the human genetic code, all 3.16 billion of them.
And what they found was, well, it was fascinating, and in some cases, totally unexpected. For one thing, it turns out we have way fewer genes than scientists predicted, only about 20 to 25,000. But here's the real stunner. The DNA of any two people on this planet is 99.9% identical. Just think about that. And here's another shocker for you. Of all that DNA, less than 2% of it actually contains the codes for making proteins. For years, scientists called the other 98% junk DNA, but we know now it's anything but junk. It's doing all sorts of critical jobs, like regulating when genes are turned on or off. It's the operating system, not just the apps.
Okay, but wait a second. That brings up a really good question, right? If we're all 99.9% genetically identical, how on earth can a crime lab use DNA to pinpoint one specific person out of billions? Well, the secret is to completely ignore the 99.9% that's the same and zoom in on that tiny, tiny 0.1% that's different. And that technique is what we call DNA fingerprinting. And it's clever because it doesn't even look at our genes, the parts that code for eye color or height. Instead, it focuses on these other stretches of DNA that are full of repeating patterns. And these are the areas of our genome that are super variable from person to person.
So let's walk through a classic example. A crime is committed. The forensic team finds a biological sample at the scene. Could be blood, hair, anything with cells. They extract the DNA from that sample, and then they compare its fingerprint to the DNA fingerprints of a couple of individuals.
What they get is this unique pattern of bands, kind of like a genetic barcode that's unique to each person. And it's pretty clear, right? The pattern from the crime scene DNA is a dead-on match for individual B, but it looks nothing like the pattern for individual A. It's an unbelievably powerful tool. So when you boil it all down, what's the big takeaway here? It's that DNA is so much more than just a molecule. It's an ancient history book. It's a detailed instruction manual. It's the epic story of life itself, passed down for billions of years, and it's all written in a simple four-letter alphabet. And that leaves us with one last massive thought to chew on. For pretty much all of human history, all we could do was wonder what was in this book of life. But now we can read it. And with tools like CRISPR, we're starting to learn how to rewrite it. The question isn't, can we anymore? The question is, should we? And what on earth happens next?